Welcome, student, in the new session of biotechnology and its application. Biotechnology, its application we can find in different areas, in different fields, like in pharmaceuticals, therapeutics, diagnosis, genetically modified crops, for the production of processed food, in bioremediation, for waste treatment, and for energy production. There are three critical research areas of biotechnology. Number one is to provide the best catalyst in the form of improved organism, usually a microbe or a pure enzyme. The second area is to create an optimal conditions through engineering for a catalyst to act on it. And the third, last important research area is downstream processing technologies for the purification of the product or the protein that is produced using this technology. Let's begin with the application in the field of agriculture. There are three options for increasing food production. Number one is agrochemical based agriculturing, second is organic agriculture, and third is genetically engineered crop based agriculture. A genetically modified organism are the plants, bacteria, fungi, and animals whose genes are altered by manipulation. The advantage of biotechnology, that is genetic modification in plant, is as follows. It makes crops more tolerant to abiotic stresses. Pest-resistant crops reduce use of chemical pesticide. It also helps to reduce post-harvest losses. It increases efficiency of mineral usage by plants so that to prevent early exhaustion of soil fertility. It also enhances nutritional value of food, for example, golden rice that are rich in vitamin A. GM is used to create tailor-made plants to supply alternate resources like starch, fats, pharmaceuticals, etc., fuels to industries. Let's learn about the pest-resistant plant. A pest-resistant plant acts as a biopesticide. It reduces the need of insecticides, which are chemical, which are harmful. Examples are BT cotton, BT corn, rice, tomato, potato, soybean, etc. BT cotton. Some strains of Bacillus thuringiensis have proteins that kill insects like solipterans, which is a beetle, lepidopterans, tobacco birdworm, armyworm, and dipterans, which are fleas and mosquitoes. Bacillus thuringiensis forms a toxic insecticidal protein, which is Bt toxin crystal, during a particular phase of their growth. It does not kill the bacillus as it exists in an inactive protoxin. But when an insect injects an inactive toxin, that converts into an active toxin due to the alkaline pH which is present in the gut of bacteria, that worm, right? And it solubilizes the crystal. The toxic binds to the surface of midgut epithelium and creates pore. It causes cell swelling and ultimately it leads to the death of insect. Bt toxic genes were isolated from that bacteria named as Bacillus thuringiensis and was incorporated into crop plants such as cotton. Most Bt toxins are insect group specific. The toxic is coded by gene Cry1AC named as Cry. An example is given protein encoded by genes Cry1AC and Cry2AB control cotton bollworms, whereas Cry1AB controls corn borer. The next one is nematode resistant in tobacco plants. A nematode, that is Melatogyne incognita, infects the roots of tobacco plants, which causes a reduction in the yield. It can be prevented by RNA interference, which is RNAi strategy. RNAi is a method of cellular defense in all eukaryotes. It prevents translation of a specific mRNA, which is needed for splicing, due to complementary double-stranded RNA molecules. The source of complementary RNA is from an infection by RNA viruses or mobile genetic elements which are known as transposons that replicate via RNA intermediary. Using agrobacterium vectors, nematode-specific gene is introduced into the host plants. It produces both the sense and antisense RNA in host cell. These RNAs are complementary, so they will form a double-stranded RNA. It initiates RNAi and slices the specific mRNA of nematode. Thus, the parasite cannot survive in a transgenic host expressing specific interfering RNA. That was it about the application of biotechnology in the field of agriculture. Now, the application of biotechnology in the field of medicine. Recombinant DNA technology helps for the mass production of safe and more effective therapeutic drugs. The products from non-human sources induces unwanted immunological responses. But a recombinant therapeutics does not have such problems. At present, about 30 recombinant therapeutics have been approved. Of these, 12 are being marketed in India. We will learn three applications of biotechnology in, in medicines. A genetically engineered insulin, second one is gene therapy, and third one is molecular diagnosis. Let's begin with the genetically engineered insulin. Insulin is used to manage adult onset diabetes. Insulin from the pancreas of animal causes allergy or other types of reactions to the foreign proteins. Now, it is possible to produce human insulin using bacteria. Insulin consists of two short polypeptide chains, chain A and chain B, that are linked by disulfide bridges. In mammals, insulin is synthesized as a prohormone. It needs processing to become mature and function hormone. Here on the screen, you can see that how the insulin, it's the structure of insulin, active and inactive insulin. So it needs processing to become mature and functional hormone. The prohormone contains an extra C stretch, that is C peptide. This is removed during maturation into insulin. In 1983, Ali Lilly, an American company, prepared two DNA sequences corresponding to A and B chains of human insulin. They were introduced into plasmids of E. coli to produce insulin chains. Chains A and B were combined by creating a disulfide bond to form human insulin. Next one is gene therapy. It is a method to correct a gene defect in a child or an embryo. Here, genes are inserted into a person's cells and tissues to treat a hereditary disease. It compensates for the non-functional gene. First clinical gene therapy in 1990 was given to a 4-year-old girl with a deficiency of ADA, that is adenosine diaminase. It is caused due to deletion of the gene for adenosine diaminase, the enzyme for immune system to function. Treatment by bone marrow transplantation or enzyme replacement therapy is not completely curative, so in that case, gene therapy is more effective comparing to any other kind of genetic disorders. A gene therapy for ADA deficiency is given. A flowchart of ADA deficiency gene therapy is given. Collect lymphocytes from the patient's blood and that is grown in the culture. Then, in that culture, introduce a functional ADA complementary DNA into lymphocyte using a retroviral vector. Then they were returned to the patient. This should be periodically repeated as lymphocytes are not immortal. If ADA gene from bone marrow cell is introduced into cells at early embryonic stages, it could be permanently cured. The third application of biotechnology in medicine is molecular diagnosis. A conventional methods are not suitable for early diagnosis of disease. It is possible by techniques like recombinant DNA technology, PCR, and ELISA. PCR. Presence of pathogen is normally suspected only based on symptoms. By this, the concentration of pathogen is already very high in the body. However, very low concentration of bacteria or virus can be detected by amplification of their nucleic acids by PCR. The uses of PCR to detect HIV in suspected AIDS patient, to detect mutation in genes in cancer patient, and to identify many other genetic disorders. A single-stranded DNA or RNA which is tagged with a radioactive molecule probes is hybridized to its complementary DNA in a clone of genes. Then it is followed by detection using autoradiography. So the clone having mutated gene will not appear on photographic film, but because the probe will not have complementary with the mutated gene, and this is how we can detect the defect in genes. The other one, molecular diagnosis, is ELISA, which is enzyme-linked immunosorbent assay. ELISA is based on principles of antigen-antibody interaction. Infection by pathogen can be detected by the presence of antigens, that is proteins, glycoproteins, etc., or by detecting the antibodies synthesized against the pathogen in the body. So that was it. That's what it is about the application of biotechnology in the field of medicines and agriculture. Thanks for watching.